Hi everyone, welcome to my channel. My name is Lorenzo and today we're going to look at GPCRs, an overview. So we're going to look at what are GPCRs, what pathways they are involved in, and the structure and function of these receptors. In the next videos we're going to look at the pathway here in pink, so the adenylyl cyclase cyclic AMP protein kinase A that involves GPCRs, and phospholipase C deacylglycerol inositol 3-phosphate pathway. So let's start by looking at the structure of GPCRs. So as you can see in these pictures, GPCRs are composed of 7 transmembrane domains. So they are 7 transmembrane domains. And the transmembrane domains are composed of an alpha helix that I've drawn here, that is hydrophobic, so let's say hydrophobic, and a loop that's hydrophilic. And this makes sense because it's a receptor that's embedded inside the membrane, so it needs to have a part that is hydrophobic and a part that is hydrophilic. And this part is repeated 7 times, so 1, 2, 3, 4, 5, 6, 7. And we have the N-terminal domain outside the cell and the C-terminal domain inside the cell. And in the C-terminal domain there are these proteins that are called G proteins. In fact, G protein couple receptor is named like that because it's coupled with G proteins. And the G proteins are mainly multimeric proteins, so they are made of different subunits, and we have, for example, G alpha, G beta, and G gamma. And these are the main subunits, so it's a trimeric protein. We have 3 subunits inside this G protein. And why is it called G protein? Well, because if we look at the alpha subunit, that is this subunit, here we have a little scheme of the alpha subunit, and the alpha subunit can be bound to GTP or GDP. So if it is bound to GDP, it's inactive, and if it's bound to GTP, it is active. And so it's a G protein because it is associated with GTP or GDP, so guanosine diphosphate or guanosine triphosphate. And so this is the inactive state, and this is the active state. How do I remember that? Well, GTP has 3 phosphates, so it has more energy, while GDP has less energy because it has only 2 phosphates. And so GTP, that's higher energy, is active, while GDP, that has lower energy, is inactive. And to transition from an inactive to an active state, we need to exchange a nucleotide. So here is an exchange, because we cannot add phosphate to GDP to transform it into GTP inside the protein, while to go from active to inactive, we just need to hydrolyze the GTP to GTP plus phosphate. So this is the mechanism. And the G proteins are mainly for hormones, for example, norepinephrine, or neurotransmitters, for example, dopamine or glutamate. In fact, the GABA receptor, for example, or GABA, well, yes, GABA, the GABA receptor that's a target for different drugs, is G protein couple receptor. So, for example, benzodiazepines or barbiturates bind to the G protein couple receptor. Let's take another page. They bind to a G protein couple receptor, such as this one, and they activate different proteins inside the cell on the neuron, and they can also trigger channels to open and close. So this is a discussion for another video that I will make about different type of G protein kappa receptor and the pathway that they are involved in. So for example, here we have adenylyl cyclase and cyclic AMP proteakinase A. This is a quite simple, and mainly these exams and courses focus on this pathway, for example. And so we have the G alpha subunit, that's the main protein that's associated with the G protein kappa receptor. And we also have, for example, adrenergic receptors. That's another target for some medicines. So for example, beta agonists, and other agonists of the adrenergic receptor bind to a G protein kappa receptor. So these are very, very, very important. We also have, for example, in our eyes, we can see in the dark because we have a G protein kappa receptors that are bound to a derivative of vitamin A, so retinol that isomerizes. But as I said, I will make videos about all these pathways in the future. So if you are interested in this, please check out my channel and maybe also subscribe to support it. Thank you. I will see you in the next one. Bye bye.